Okie dokie, guys, we're going to go ahead and go over 3.3, which is going to be environmental impacts on enzyme function. The basic gist behind this is that when it comes down to your enzymes, they prefer to work in specific environments, right? We know that we can change the salinity of certain solutions, we can change the temperature of solutions, and I mean, when we say solutions, just environments in general. So basically, more or less, these enzymes are structured to work in specific areas or in specific environments. And then once you start to change up the environment, then they just stop working as intended. So what you can see here is that this just talks about the process in which we will start to denature enzymes. Denaturing just means that the protein is losing its shape. And again, right, structure determines function. If it loses its shape, it loses its function, and then your enzyme isn't able to work as properly anymore. This is actually kind of important because you'll have certain enzymes like amylase that can be found in both like your saliva and I believe in your small intestines. Gotta just double check that real fast. But you have like these carbohydrate or these maybe it's like protease. You have like these protein enzymes that are found like within like your stomach acid, right? And then like with your intestines. And basically those environments have two different pHs, but they are both breaking down proteins, right? So the idea is that, hey, I have an enzyme that will work perfectly fine in this environment and an enzyme that will work perfectly fine in this other environment. But then the problem ends up being is that the problem ends up being is that if I were to throw like the one that doesn't like the acidic environment into the stomach, well, then it's not going to be able to work properly anymore. So that's all the stuff in there. There is a relevant equation, the pH minus log for the hydrogen ion concentration. I'm going to be honest with you. I've never seen this show up anywhere. I'm not going to go spend that much time into that. All right. So a little bit of blast from the past, right? So we need to go talk about our proteins, right? Keep in mind, right? Your primary structure, right? Your primary amino acid sequence is going to have some kind of folds that take place, right? So this is going to be like your alpha helixes, your beta sheets, right? So we have them right there, and eventually it's going to go get that three-dimensional fold, and then we can end up having quaternary structures where multiple folds all come together, right? Now keep in mind these guys like are they'll have like hydrophobic interactions, or they'll have hydrophilic interactions, or they'll have ionic interactions and stuff like that, right? The intent or the idea behind this is that if I were to put in, we're like they like to be in cytosol, right? Like cytosol has like a specific salt content and sugar content and pH and all this other nonsense, right? And different parts of your body will have like different environmental impacts. Well, if the protein is put into an environment that it likes, it's going to go work perfectly fine. But then if I put it into an environment that may be completely different, then it's not going to go work. And that's really the whole shtick of what we're going to be taking a look at in here. Okay. This is going to be talking about denaturization. So you can go watch a video about this, but denaturization literally just means is that the protein shape becomes unfolded and then it's unable to do its job. Why? So like conceptually what's happening, right? Remember that we have like these folds because there might be a disulfide bridge or hydrogen bonding or hydrophobic hydrophilic interactions, what have you. Basically what you're doing is that you change the outside, right? So let's just say if this is like a pH of 7 to begin with, right? And we say like the pH is neutral to begin with, but then I end up putting us into like an acidic environment. Sorry, I'm like deleting my stuff by accident. Let's say I put myself into an acidic environment, and now I'm like at a pH of like 5. Well, now these certain interactions are going to go break apart, and they break apart because they're now like fighting with like the hydrogen ions that are all accumulating in this area now. And then that ends up causing it to misfold. The exact same thing ends up happening if we have basic, right? If it's a like a pH of like 11 and we're basic, well, instead of it hydrogen ions, it's hydroxide ions, hydroxide. And basically those are now the things that are interacting with, right? Whether it's a positive sign or a negative sign, they're both going to be affecting the protein in a specific way because certain things may be attracted to this negative ion, or they could be attracted to this positive ion, or they could be repelled by it, right? And they're trying to stay away from it. Because you put two positives near each other, they will repel away from each other. And basically, an accumulation of all this nonsense happening is going to end up causing this unfolding to take place. We can also heat it up. The idea here is that if you, let me go ahead and just reset this right for a second. If we wanted to say, like, we wanted to apply heat, right? Geez, I cannot spell right now. If I wanted to apply heat, right? Well, what heat does is that it's increasing the kinetic energy of atoms and molecules. Like that's literally like what temperature is. It's like the average movement of the stuff, right? You should have learned this in chemistry. If you haven't, there's a little chemistry thing, right? So what we're doing is that we're increasing kinetic energy. These atoms are vibrating a little bit more and more and more and more, and because they're vibrating, right? If I had like an O and an H, right? And I was like doing a hydrogen bond. Well, because they're vibrating, maybe this one shifts down and this one shifts up. That causes this hydrogen bond to break, and then it starts to denature that way. So that's like the mechanics of what's happening here. So whether you change up the environment, like the pH, you can also change up the salinity. You can change up the temperature. Just changing up these different things can end up having some um, dire consequences to your enzymes. All right, and that's what we're just saying here, right? Changing the environment, temperature, and pH. So this is what's called like a reaction graph. This basically just kind of shows you like how an enzyme behaves, right? So we're at like 0 degrees Celsius, right? That's 32 degrees Fahrenheit. It's freezing. It's cold. It's like no bueno, right? You are this little blue line, right? You're 37 degrees Celsius, which is like around like 96 degrees Fahrenheit. As you get more and more and more, like this is going to be like more like 110 or what have you, right? I don't have a calculator off the top of my head to know the numbers, but just kind of follow me with it, right? Um, Basically what is happening here, right? So 110 should be like 110 Fahrenheit and like they're 96, right? So what happens is that this enzyme, the enzyme activity increases as we go up, right? So when you're at 37 degrees Fahrenheit, like right here, like the enzyme is working at its peak performance, right? It is folded correctly, right? It's folded correctly. It's the right temperature, so like the folds are staying intact. There's no other nonsense that's happening in there, right? Well, then once I start to like increase or decrease the temperature, then what happens is that it causes the enzyme activity to decrease, right? And this is actually the denaturization taking place. Is that where it's losing its shape, and as it loses its shape, it loses its function, which means that its activity like decreases and decreases and decreases. So that's what's typically happening here. Is like you're just like heating them up to the point that they don't work anymore. The structure of it is disrupted, right? That's the big thing, right? The structure is being disrupted, and then it's losing its efficiency, right? It's stopped completely, or its efficiency is decreased. Okay. There are certain cases that you can end up reversing this. We can also change the shape of enzymes to get them back to where they need to go. So actually, what's neat is that temperature, you can kind of renature them to a specific point. This is why maybe like if you're having a fever, your enzymes, you may not have a good time like digesting or doing stuff like in your head because you have a relatively higher temperature. But like once your fever breaks, like the enzymes will be able to work perfectly fine. So lots of crazy nonsense like with that. We got a dark cow simulation that we're eventually going to go ahead and go do and show you with this. But what we'll be able to do is actually show you specifically where certain enzymes will work perfectly. So for example, if you go take a look at the board right here, you're going to go and find that we have like pepsin. Okay, now pepsin works best in a pH of 2. So I want you to think, where do you think this enzyme is located within your body? The most acidic area, right? This is going to be your stomach, right? So your stomach is going to be where this enzyme pepsin works perfectly fine. Like it works super, super well in your stomach. It's designed that way. When we start talking about evolution, it evolved in a way for it to work that way, right? Now, if I were to go and take a look at this salivary amylase, right? Well, think about it. It says salivary amylase. Where do you think this salivary amylase is located? This is in your mouth. All right, you have this amylase that's just kind of this enzyme that's hanging out in your mouth, and if you think about it, right, like it's your mouth is not acidic. You don't actually taste like the acid that's in there, and then you're able to go and kind of pre-digest large carbohydrates already in your mouth. So what's neat is like it mixes with like spit, saliva and the enzyme, and it turns into like a slurry mush, which goes into your stomach. And then, the carbohydrates are already starting to be digested. Then you get this pepsin that will then start to digest the proteins. And then once you go into the small intestines, I'm going to introduce lipase to start to break down the lipids. And it's a really kind of neat process on how like everything gets broken down at different areas. So, um, this is something that you just want to kind of be familiar with, is how hydrogen ions relate back to pH. This is actually a reoccurring theme that shows up over here over and over and over again. If you're not really too sure about how the pH scale works, I'm going to kind of give you like a quick and dirty of it. pH 7, you have acids over here, you have bases over here. Okay. So like 5, 3, right? You go lower, you get more acidic. And then when you get higher, it's basic, right? 14 is the highest, and like, 1's like the lowest, right? So, um, now, when it comes down to neutrality, your neutral molecule is going to be water. Now, what's important about water is that water, if we wanted to break it down into ions, if you kind of follow the logic here, I can break it down to a hydrogen ion and a hydroxide ion. So if you add the H plus and the OH minus, we would actually be able to get an H2O molecule. And basically what happens, it's like powers of 10. So this is like 10 to the 1, 10 to the 2, 10 to the 3. And it's always talking about the hydrogen ion concentration. So this is where it kind of gets like really kind of weird, because it's like 10 to the minus 1 here, 10 to the minus 2, and I don't particularly like that, but it makes sense if you kind of wrap your head around it. So we'll do the acids first and talk about it, right? So 10 to 1 basically means for every H2O, you're going to have 10 hydrogen ions, and that's going to be specifically there. Let's do this one in a different color so that way you can kind of like follow like the logic of what's happening, right? Now, if I'm doing like 10 to the 2, that basically means that for every H2O that I have, I'm going to have 100 hydrogen ions, right? And then 10 to the 3 is going to be 1,000, 10 to the 4, 10,000, right? So like you can kind of like follow like the math behind it, and that's how that ends up going, right? Um, if we go in the opposite direction, this is where people kind of get confused. And it's kind of like it's designed this way to be a little bit weird. So I'll try to slow it down, right? As we go this way, we're increasing the hydroxides, and here's why. The idea is that for here, it's like every 10 H2Os, there's now one H plus. Then it's like 100 H2O for an H plus. And the thing is that you need an H plus and an OH minus to make the water. So instead of it being like these H pluses are accumulating on this side, it's actually more OH minuses. And it's like all these OH minuses. So as I'll redraw the thing so you can kind of like understand one more time, right? For your pH scale, right? Here's 7. As we go further in this way, the amount of OHs increases like more and more and more, right? And then it's like, you can kind of like use your imagination. Like there's lots of OHs, like when we start going over to this side. And then when we go over to the opposite side, right, you'll get more H pluses as you go this way, right? Like more H pluses, more H pluses, a lot more H pluses. This kind of goes with the idea that you would actually like know this up ahead of time. I know that not everybody is clear on this, so if you have questions on this, just let me know. The only reason why I bring this up is that hydrogen ion specifically concentration will come back up for cell respiration and photosynthesis, uh, specifically like certain regions of where these processes take place. There's like a huge accumulation of these hydrogen ions. So it's like they're very acidic environments, and it's kind of like a weird like, hey, can you connect the dots? and you don't even know what dots are even there to connect. So if this didn't make sense, like feel free to just kind of ask questions next time we're in class, and I'll try to have another picture available, and maybe I can explain it one more time for those who weren't really too sure. But anyway, we're going to get back into it, right? So we're increasing the hydrogen ions, we're increasing the hydroxide ions. More or less of them is going to change the shape of the protein, all right? There's other things that we can end up doing that we can take a look at, right? So we can end up looking at adding enzymes versus no enzymes. So this graph basically is talking about reaction rate, right? So the core, like how many reactions, right? How many reactions versus the concentration of substrate, right? So the substrate is right here is our guy, right? We're talking about the little circles, right? So you're increasing the amount of substrates, the amount of stuff that our enzyme can play with. So it should probably make sense that if you have more stuff, you're going to have more reactions, right? Like think of it like this. If you have two pieces of bread and you have, one ham, slice of ham, how many sandwiches can you make? Obviously, you can only make one sandwich, right? It's not a trick question. Well, what's going to happen if now I end up giving you, more bread and more ham? Well, you're then going to be able to make more sandwiches. And that's kind of like what's happening here. As you increase the substrates, we should start to see an increase. Now, there's a little bit of a caveat to that, which we'll show in the actual demonstration on the online simulation later on. But and you should also see this within the handout that I gave you as well. Okay, but this increasing substrates should mean more reaction. Now we can see reaction without the enzyme, it's just going to gradually go up and everything's gonna be like hunky dory, right? Because it's like, okay, cool, like the reaction will take place over time. Now, when I start to throw an enzyme into the mix, something a little bit crazier ends up happening, right? It exponentially grows because the enzyme, right, lowers the activation energy. I am using less energy to make the same reaction take place, so I can just make the reaction happen a lot more efficiently. But what you can see is that it's going to cap off even with the enzyme. There's a maximum rate that I'm going to go and handle, right? And here's a way that I've always described it, right? If I asked you how many of these ham sandwiches, how many ham sandwiches can you make within like a minute, right? I just like think about it, like two pieces of bread, one ham. Like just answer that for a second, okay? Maybe you'd be like, I can get like one sandwich every 10 seconds. I say, cool, that sounds great. Okay. So then if you can do one sandwich every 10 seconds, right? Well, if I give you one, like, the amount of stuff, you can try to follow the graph, and this might, hopefully you can connect the dots with this. Okay. Here is you doing the reaction with one, right? You can make one sandwich definitely within a minute. You can also make two sandwiches within a minute. You make three sandwiches, four sandwiches, five, and six. But here's the problem. Once you're at six, right? We already said you can make at most six sandwiches a minute. So no matter what, if I give you more sandwich material, right? More substrate. If I give you more substrate, you can't make any more sandwiches because you're only so efficient at making them. And that's why we see this reaction graph works the same way, right? The enzyme is only so efficient. The enzyme can only interact with so many substrates before it's like kind of overwhelmed and it's like, look, I can do this reaction, but I have a maximum rate I can achieve. So if you want this reaction to happen a little bit more, get more enzymes. And that's kind of like one of the reaction graphs that you should have seen, right? You can see about changing substrates, changing reactions. Um, all right, cool. So increasing temperature can also speed up chemical reactions, right? That is, gosh, kinetic movement theory. Basically, when you increase the movement of the particles that are in there, right, within the system, it increases the rate in which reaction occurs. Kind of like the same way like with diffusion. Like if you were to put Kool-Aid into a or like a lemonade powder into water, right? Well, you can pour the stuff and it'll eventually diffuse, or you can stir it in order to increase collisions, right? That's what this is referring to. It's talking about like the substrates colliding with the enzymes to be able to make more reactions taking place. But the problem is that when you increase the temperature, right? Again, our enzymes, they have this like Goldilocks spot, right? It's like it's not too cold, it's not too hot, like it's just right. But as soon as you get too high or too cold, the actual enzyme starts to not work as well. There are two other ways that we can end up doing this. We have inhibition. There are these little foam models that we're going to be playing with in class to demonstrate what inhibition is, but you have competitive and non-competitive inhibition. And there's a picture to kind of show you what that ends up looking like. Definitely write these guys down. You need to know about how this works. This is very big on cell communication, but like, if you want to listen to this, right, you can tell that this substrate can fit within this enzyme, right? But you see this guy that's like in the way that's preventing it from actually doing it. This is a competitive. This is a competitive inhibitor. And we're going to show you a picture. Competitive inhibitors bind to the active site, while what's called non-competitive, they bind allosterically or allosteric inhibition, which means that they bind somewhere else onto the enzyme to change its shape. And we're going to go show you that in a bit. But yeah, competitive means that they are on the active site, they fit in there. They're not going to make the chemical reaction occur. They're just saying, like, you can't make this happen. And basically, the whole purpose about it is that we're blocking the function of an enzyme, so that way we can turn it off. Think about the ham sandwiches, right? Do you want to make infinite ham sandwiches? I mean, I love me some ham sandwiches. I'm going to tell you a funny story. Um, Capriati's. I love me some Capriati's. I was at doing a training and we had Capriati's like 3 days in a row, and we had like Capriati's for lunch and dinner. I did not want to look at another Italian sub for like the rest of the summer. And honestly, like, they made me sick to my stomach when I like saw them. All right. So what I'm getting at though is, like, you don't need infinite ham sandwiches. You just need enough in order to go feed who needs to be fed, and then you can be done with it, right? So the idea about these inhibitors is that it just basically blocks the function from happening, right? And when we start talking about cell communication, we can basically put these inhibitors on or take these inhibitors off in order to drive a cell function to occur. It's actually pretty cool. It's basically your cells, like, it's cell communication, but your cells are just basically, uh, realizing what they need to go and do, right? If you're hungry, you go and find food. If you're not hungry, you're not going to go find food. And that's basically how it's done at the cellular level. Like, if I don't need energy, I'm not going to go break this stuff down. This is just showing you the idea about how some of these inhibitors work. So here we have we'll look at option A to begin with. This is an inactive enzyme with an activator. So this is not going to be inhibition, like it's kind of turned off, but this activator, what it does is that it will bind to the enzyme, changing the shape, allowing the substrate then to form, and then we can actually have it take place, right? And then this will be in it's called an on position until I break down all the substrates there. And then once all of them done, this guy can go leave, and then we turn the enzyme off. This one right here is that we can have an inactive enzyme, and then we can take a protein kinase, and we are going to phosphorylize. Really, really cool thing. Basically, you stick a phosphate group onto an enzyme and it changes the shape of it. And what, again, right, your phosphate groups is a phosphate with a bunch of oxygens on it. This thing is like super, super negative all over the place. All that negatives attached to the enzyme is going to go change its shape, and then now the substrate's able to go ahead and bind to it. So these are kind of like two different ways that we can turn them on, right? This is not inhibition, but this is just basically how enzymes be turned on and turned off. This is showing you competitive inhibition and non-competitive inhibition. Competitive, right? The competitive inhibitor will bind to the active site, making it so the substrate can't go on there. So the substrate kind of hangs around and, like, does whatever. And non-competitive, right? It can be a non-competitive will go into the spot, changing the shape of the protein, and now it prevents the substrate from being able to bind in there. Now, something that I'd like for you to go do is just, I'll delete this and kind of show you. I want you to see the difference between the inhibition versus the activation, right? It's kind of like they're opposites of each other. Here, it is off, and then I turn it on with an activator, right? Versus here, it's on, and then I turn it off with the inhibitor, right? So just kind of be aware of that. Devil's in the details, and there's that. Um, this is just showing you a little bit of reaction graphs. You can probably kind of make sense of like a normal enzyme with a competitive inhibitor, non-competitive inhibitor. You can kind of see about how it ends up changing the rates of reaction. It should just make sense, right? And a competitive inhibitor, the idea here is that I can maybe still have some of the reactions occurring. Um, it's just, the substrate, like, sorry, the non-competitive inhibitor like falls off. Like, all right, cool, I can have the reaction. The enzyme still has a bit of a shape still there that maybe the substrate can bind to a little part of it and still have the reaction occur. It's just not nearly as effective. Non-competitive, this one's way, 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 way more efficient because it's going to legitimately change the whole shape of the protein. And when you change the whole shape of the protein, you might have some reactions, but it's going to be really, really difficult. All right, folks, that was it. Make sure to get that enzyme and cellular regulation handout completed. The model one was the two digestive enzymes. So you got pepsin and lipase. You're going to go ahead and just look at them in different pH environments. You should be able to follow with what's happening after looking at the notes. And then model two is amylase and rate of reaction by changing different environmental conditions, temperature, enzyme concentration, and substrate concentration. So please go ahead and make sure you get those all done. Really, we're going to be going over questions 1 through 19. I'd love to go through the extension questions, but don't feel like you need to go and knock them out, okay? All right, I will catch you on the next one. Take care, everybody. Bye.